What if I told you that a hacker could take over your computer using nothing but a strand of DNA? Sounds crazy, right? Like something out of a sci-fi movie. But this isn't fiction. In 2017, researchers at the University of Washington actually did it. They successfully encoded malicious software into a physical strand of DNA and then used it to gain control of the computer that analysed it. But why would anyone even think to do this? And more importantly, how does a DNA molecule become a cyber weapon? Well, DNA sequencing has exploded over the past couple decades. We use it for everything: medicine, ancestry testing, crime investigations, and even agriculture. But here's the thing, all this genetic data has to be processed by computers, and that's where our story begins. The researchers at Washington had a disturbing thought. What if the software analysing DNA sequences wasn't as secure as we assumed? Could someone potentially hide malicious code within a DNA sample itself? To answer this question, we need to understand three key concepts: how DNA is processed and stored on computers, how a buffer overflow attack works, and how DNA synthesis lets us create malicious DNA sequences. Let's break down each one. DNA is made up of 4 nucleotides: adenine, thymine, cytosine, and guanine. These 4 letters form a natural code that carries the instructions for building every living thing on Earth. When DNA is sequenced, it goes through multiple stages of processing. First, scientists extract the DNA sample from cells, then fragment it into smaller pieces, and amplify these fragments using PCR to get enough material for analysis. Next, these fragments run through a sequencing machine, which reads the genetic code and produces raw data files. The raw data then undergoes downstream processing like quality control, alignment to reference genomes, and variant calling to make biological sense of the sequence. The sequencing machine typically outputs what's called a FASTQ file, or BCL files, which are then converted to FASTQ files. And these files contain not just the DNA sequence reads, but also quality scores for each base, which indicate how confident we were that they were called correctly, as well as additional metadata. While we see this as plain text, computers actually store everything in binary code. Initially, the FASTQ file uses the ASCII system, which assigns each letter, number, and symbol in the English language a special binary code. For example, here's the ASCII code for the sequence 𝗖𝗶𝘁𝗵. But this ASCII encoding is actually very inefficient for storing DNA data. This is because the ASCII system was designed to represent many different characters, including letters, numbers, and symbols, while DNA sequencing data only needs 4, 𝗖𝗶𝘁𝗵, or 5 if you count the ambiguous space n. That means we're wasting significant storage space. This is why the sequencing data in these files are often compressed using a 2-bit encoding system. This compression reduces the storage requirement by 75%, from 8 bits per base to just 2 bits. The quality scores and metadata don't use this 2-bit encoding compression, as they have more letters and symbols and so leverage other techniques, which is not relevant for this video. This sequence compression process, converting the ASCII encoded DNA sequence in a FASTQ file into the efficient 2-bit format, is exactly where our researchers found their opportunity. They exploited a flaw in a compression tool to trigger the release of malicious code onto an unsuspecting computer which was attempting the compression. So what exactly was this flaw? Well, to understand this, we first need to understand the specific hacking methods used by the researchers: buffer overflows and reverse shell payloads. Let's start with buffer overflows. When a program runs, it allocates specific amounts of memory, called buffers, to store different pieces of data. These buffers have defined boundaries. If the program receives more data than a buffer can hold and doesn't properly check the data size, the excess data will spill beyond the buffer's boundaries, overriding adjacent memory. In properly designed software, the program would check if the incoming data fits within the allocated space and reject oversized inputs. But when these checks are missing or flawed, hackers can exploit the overflow to inject their own instructions. In a computer's memory, things are organized in a specific structure when a program runs. This includes the program's code, variables, and special memory addresses called return addresses. A return address tells the processor where to go next after completing a function, essentially a bookmark for the program's execution flow. The researchers targeted 𝗖𝗶𝘁𝗵, a program designed to compress FASTQ files. Now, it's worth noting that the researchers did actually modify this tool on the host computer to make it more vulnerable to attack and improve their chances of success. They basically removed the limit on input read lengths, making it vulnerable to buffer overflow attacks. They also modified some common security protections on the test computer, which you can read about in the paper linked below. But essentially, these system-wide modifications made the attack more likely to succeed and are quite typical actually in security research to demonstrate proof of concept attacks. But despite these control conditions, they emphasize that similar vulnerabilities exist in real-world bioinformatics software. By carefully crafting a DNA sequence that would produce a specific pattern in a FASTQ file, they created input that would trigger a buffer overflow during the compression process. The critical moment occurred when 𝗖𝗶𝘁𝗵 attempted to compress the ASCII encoded FASTQ data into the efficient 2-bit format. During this compression, the excess data spilled over and overwrote the return address in memory. This is where the real magic happens. Instead of returning to its normal routine, the program jumped to the researcher's malicious code. The CPU doesn't distinguish between legitimate program instructions and malicious code. It simply executes whatever instructions it's directed to. What did this malicious code do? The researchers designed a reverse shell payload, an attack that turns the tables on normal hacking approaches. Typically, hackers try to connect to their victims' computers. A reverse shell flips this model. It makes the victim's computer initiate a connection back to the attacker. This is particularly effective because it often bypasses firewalls, which are typically configured to block incoming connections but allow outgoing ones. The code that they encoded in the DNA executes a command that created this reverse connection using bash, a command line tool commonly used on Linux systems. The specific command looks something like this. Breaking this down, the bin-sh-i launches an interactive shell on the victim's computer. This allows the attacker to type commands as if they were directly at the victim's keyboard. And this second part is the crucial part. It redirects all communication, both the commands the attacker types and the responses from the victim's computer, through a network connection to the attacker's computer. The connection is established to the attacker's computer at the specified IP address and port. Once executed, this creates a two-way communication channel. The attacker can send commands to the compromised computer and receive the results, giving them complete control over the system. They could potentially access files, steal data, install additional malware, or use that computer as a jumping off point to attack other systems. So that's all great and all, but there's one more part to this story. How did the researchers actually encode their malicious code into a physical DNA strand? Here's the detailed process the researchers used to create their attack. They started with their reverse shell payload. When you execute this command on a computer, it actually converts it to something called machine code, which is the raw binary language that computers understand directly. So the researchers needed to first convert the text to machine code before reverse engineering the DNA sequence. Now, they couldn't just have the reverse shell command on its own. Before their actual attack code, they added what's called a NOP sled. Imagine it like a slide that leads right to their malicious code. NOP stands for no operation. It's an instruction that tells the computer do nothing and move to the next instruction. Why add instructions that just do nothing? Well, when trying to overflow the memory buffer, it's hard to know exactly where the computer will land when it starts executing code. The NOP sled ensures that no matter where it lands in this region, it will harmlessly slide down until it hits the actual attack code. It's like putting a giant target around their bullseye. Now for the magic. They needed to convert all of this computer code into DNA letters, A, T, C, and G. Remember how DNA bases get converted into binary during compression? Well, working backwards, they could figure out exactly what DNA sequence would be needed to produce their desired machine code after compression. So the DNA sequence was structured like this. It started with the no operation sled, then it had the return address pointer, and the malicious reverse shell payload at the end of the sequence. When this carefully crafted DNA sequence is processed, a FASTQ file is produced with the DNA bases in ASCII text format. Next, the vulnerable 𝗖cie_HIJACKed program tries to compress this data by converting from ASCII to 2-bit encoding. During this conversion, the program writes beyond its allocated buffer, what's known as a buffer overflow. This overflow allows the attacker to control the program's execution by overwriting the return address. When the 𝗖cie_HIJACKed function tries to return to a safe place after execution, it instead redirects to the NOP sled, which leads directly to the malicious shellcode. The CPU executes the binary instructions, establishing a remote connection to the attacker's system. The brilliance of this attack is that the researchers calculated exactly what DNA sequence would produce their desired machine code through this specific processing pipeline. But it should be noted that creating malicious DNA wasn't as straightforward as it might seem. You can't just combine DNA bases in any order. There are several biological roadblocks that had to be considered. First, DNA stability. Not every sequence of bases translates into a stable DNA molecule. Some sequences with long homopolymer regions, like lots of A's or G's, lead to errors during synthesis and sequencing. Other sequences were too repetitive, making them difficult for the sequencer to read accurately. Then there was the GC content. That's the percentage of G and C nucleotides in the DNA. DNA with too few G's and C's is less stable, especially at the beginning of the sequence. The researchers needed to keep the GC content balanced, aiming for roughly 40 to 60% throughout the strand. And finally, there were length limitations. Commercial DNA synthesis services have limits on how long a DNA strand can be, typically around 200 to 300 nucleotides for accurate results. It was only after considerable trial and error, testing different encoding approaches, that the researchers finally created a 176 base pair DNA sequence that ticked all the boxes. It was biologically stable, meaning it could be reliably synthesized and sequenced, and it was functionally malicious, meaning it would execute their reverse shell payload attack when processed by the vulnerable software. And if you're wondering how you actually make the physical synthetic DNA strand, well, you can literally order custom-made DNA online from a commercial DNA synthesis company. Companies routinely synthesize DNA for research purposes. You just send them the sequence you want, and then they'll mail you the actual physical molecules. The fascinating part about this experiment is that the attack crossed the boundary between the physical and digital worlds in a way that had never been demonstrated before. The attacker creates malicious code and converts it to binary machine code. That binary is then translated into a DNA sequence using a 2-bit encoding system. The DNA sequence is sent to a commercial synthesis company which produces a physical DNA strand. A lab sequences the DNA, turning it into a digital FASTQ file. The FASTQ file is processed by the 𝗄𝗶𝘁𝗵 software, which attempts to compress it, and during compression, a buffer overflow occurs, allowing the malicious payload to execute. The system unknowingly runs the attacker's code, giving them remote access. It's like hiding a computer virus inside a biological molecule and then having the computer itself reconstruct the virus when it tries to read the molecule. So now that we've seen how researchers encoded a computer attack into a DNA strand, let's talk about what this actually means. Once attackers gain control through this DNA encoded exploit, what could they do? They could steal sensitive patient data from the host computer, they could install malware like keyloggers which steal passwords, or deploy ransomware, and they could even use the compromised computer to attack other systems on the same network. Pretty scary stuff. But before you start worrying about the evil scientists hacking into your computer while you're analysing DNA, let's put this into perspective. This wasn't a real-world cyber attack. It was a controlled experiment conducted under specific conditions. The researchers disabled several standard security features on the target computer and modified the compression program to make it more vulnerable. That said, they did find over 24 similar vulnerabilities in the original unmodified program. This suggests that while they made their attack easier, similar attacks could potentially work against regular bioinformatics software. And this brings us to perhaps the most alarming finding from the research: just how insecure bioinformatics software is compared to other types of programs. The researchers found that at the time of conducting the experiment in 2017, many bioinformatics tools had serious security flaws. This figure from the paper shows significantly more insecure function usage in software involved in next-generation sequencing applications compared to regular software. This basically means that some code in bioinformatics programs includes things like functions that don't check memory limits, lack proper bounds checking, and leave systems vulnerable to data overflow. Looking at figure 5 from the paper, we can see exactly the kind of issues that can lead to overflow attacks like the one they demonstrated. In some cases, they even found revealing comments in the code such as max sequence length 2000 should be enough for now, indicating that security considerations weren't a primary concern when these tools were developed. So why is bioinformatics software so vulnerable? Well, it's often developed by scientists rather than software engineers. The focus has always been on accuracy and scientific results, not cybersecurity. Until now, DNA processing wasn't widely seen as a security risk, so testing was rarely a priority. Since this research was published in 2017, the bioinformatics community has made some progress. Security awareness has increased significantly. Newer tools are increasingly developed with security in mind. However, many older, vulnerable tools are still widely used, and the rapid pace of biological research means security sometimes remains an afterthought. So there you have it, a fascinating demonstration of how a DNA molecule can act as a vehicle to take over your computer, and a stark wake-up call that security is something that needs to be taken more seriously in the field. If you want to know more about this experiment, then check out the research paper linked below, and like and subscribe while you're down there too. Bye.